Let that texture that. Mhm.
Good evening, Pratham. Yes, I'm Rahul sir. And last time we had conversed about the specificities of neuroglia, the types of neuroglia present in the CNS. We had talked about the star-shaped astrocytes. Then we had talked about the oligodendrocytes, which produces or which are responsible for producing the myelin sheath in the CNS, and it has multiple processes which can wrap around different neurons, more than one neurons, and it does the padding stuff, multiple wrapping. Good evening, Gayatri. So that's how it does it. It is smaller than an astrocyte and it has lesser processes than an astrocyte compared to the oligodendrocyte. If we compare oligodendrocyte and astrocyte, so today we'll be talking about the rest of the neuroglia and then proceeding towards neurotransmitter and the action potential generation in the neuron, right? So the next class of neurotransmitter, sorry, the next class of neuroglia that we'll be talking about is we have talked about oligodendrocyte, we have talked about the, you can say, astrocyte, and we had also talked about the ependymal cells, right? So now we'll be talking about the last one in the CNS, which are known as the microglia, the fourth type of neuroglia in the CNS. The first one is the astrocyte. This is a very, very significant classification. Oligodendrocyte, Then we have ependymal cells which produce the cerebrospinal fluid, ependymal cells. Then we have the microglia. So microglia are very, very specialized cells. Okay. So the last type of, or you can say these are the least numerous. Okay. These are the least numerous type of, you can say, neuroglia present in the CNS. Okay. And they are what? They are known as the phagocytes. Okay. They have slender processes with multiple fine branches. Okay. And they are mainly the phagocytes. So the most numerous is the astrocyte, and microglia once we talk about. So microglia is the least numerous. Okay, it comes in NEET examination. Pick the least numerous one. Okay. Next thing, what microglia are capable of? They are capable of phagocytosis. In the chapter cell, you might have learnt about endocytosis versus exocytosis. Now, endocytosis, if you divide, you have phagocytosis, that is cell eating. We have pinocytosis, also known as cell drinking. Then we have receptor mediated endocytosis also. Okay, via the clathrin molecules via the plasma membrane. So receptor mediated endocytosis. So these are the types of, you can say, endocytosis. Then we have exocytosis. So phagocytosis is done by especially the macrophages. I hope you have heard of the macrophages in the chapter blood, right? The monocytes, they differentiate into macrophages. So the liver has also macrophage known as Kupffer cells. The lung cells or the lungs, the tissues, the lung tissue has the macrophage. They are known as the dust cells. Same way, macrophages are integral part of the human brain also, because it needs to eat and it needs to devour the microorganisms if they have reached the brain, right? So it's a part of the immune system of the brain. So does the phagocytosis in the CNS. That is the work of microglia. So it's a type of, you can say, macrophage. It forms the immune system of the human brain, immune surveillance, you can say, which is extremely essential, because you cannot let the brain be susceptible to any kind of risk regarding the microorganism, you can say infection, right? So you must have some kind of macrophages, some kind of innate immune cells in order to clear the debris, in order to clear the microorganisms which may have got entry through the blood brain barrier into the brain. So that's the first line of defense as far as the brain is considered. Few years ago, one more thing I'll tell you, that lymphatic system that you might have heard of, the scientists never believed, because they never believed in the existence of lymphatic system in the brain. But 2 years ago, there was a great discovery in one of the researches which did show everyone that lymphatic system is present in the brain, and lymphatic system is responsible again for clearing the toxin and the debris. We use the function of lymphatic system in the, you can say, peripheral body, or you can say the entire body except the brain, right? You can call it the peripheral nervous system. So below spinal cord and cerebrospinal fluid, the lymphatic system is all around us. We have lymph nodes, right? We have lymphatic organs, primary organs, secondary lymphatic organs, right? So what we have is we have blood, we have, sorry, the bone marrow, the spleen, the mat, mucosal associated lymphoid tissue, gut associated lymphoid tissue. So many are there. So what happens is the brain also has its own lymphatic system in order to take away or drive out all the toxins, right? So that is known as glymphatic system. The lymphatic system of the brain is known as glymphatic system. So always know that our brain also has its own lymphatic system in order to drive away the toxins. Okay, the toxins must be driven away, otherwise you will have toxin accumulation, and the quality of the, you can say, recall capacity will decrease, and also the neuronal death will start happening. And sometimes due to the failure of lymphatic system, many neurons, they undergo necrosis or cell death, and that could lead to various brain diseases like Parkinson's, Alzheimer's. You might have heard of these diseases. Alzheimer is a type of dementia. Okay, the worst type of dementia, you can say, neuro. degenerative where the neurons start dying. In Parkinson's also, dopamine producing neurons they start dying. Okay, so these kinds of diseases may occur, may emanate from the dysfunction of the lymphatic system, insomnia, then depression, then aggressive behavior. Okay, so all those things can happen due to again a very minute difficulty in the brain or minute ailment you can say in the brain. Okay, so this will be concluding the types of CNS or sorry types of neuroglia in the CNS. Now we'll be deviating our attention to the types of neuroglia in the peripheral nervous system. Okay, so in the peripheral nervous system there are two types of neuroglia. Okay, number one is satellite cell, number two is the Schwann cell. Okay, so in the peripheral nervous system we have two types of neuroglia. Number one is the satellite cell, number two is the Schwann cell. Now, once we talk about satellite cells, so satellite cells they surround the neuronal cell bodies or the cyton, okay, in the specific places in the PNS which are known as ganglia. Means ganglia is the cluster of the neuronal cell bodies. So at that specific place or at that specific position or location, the satellite cells have been found to surround the neuronal cell body or the cyton. Okay, so satellite cells, once we talk about, they surround the neuronal cell bodies in the various ganglia present in the peripheral nervous system. It is a cluster of neuronal cell body only. Okay. Now, second thing, they regulate the interstitial fluid around the neurons, just like the astrocytes in the CNS. Okay, interstitial fluid means the fluid which surrounds the cells. Okay, with the fluid which is a part of the extracellular matrix. So they regulate the concentration, they regulate the ionic balance, the toxicity. Okay, the efflux of toxins from the cells to the interstitial fluid, and from interstitial fluid what has to be taken into the neuron. So you can say it will directly monitor the concentration of the interstitial fluid. Okay. Monitors the interstitial fluid present around the neurons in the PNS, peripheral nervous system. So these are the two main functions of satellite cells. Okay. Then we'll be coming on to the Schwann cell. Very important. These are the most important cells or neuroglia when it comes to PNS, Schwann cell. What they do is they go for myelination. Myelination of axons in the PNS is perpetuated by Schwann cell. Myelination of axons in the CNS is caused by the oligodendrocyte. So this is the real time you can say neat question. Okay, which cell is responsible for myelination of neurons in the PNS? Direct answer is the Schwann cell. In the CNS, oligodendrocyte. So this is a very common question. Okay, but you are not supposed to make mistake here. Okay, so Schwann cells they have a different name also. They are also known as neurolemocytes. In some questions, it is also asked what is the alternate name. So neurolemocytes. That's the alternative name for Schwann cell. Okay, they either form a thick myelin sheath, okay, or intended folds, means folds of plasma membrane around the peripheral, you can say, axons in every part of the peripheral nervous system. Okay, so what happens is you can say wherever a Schwann cell covers an axon, the outer surface of the Schwann cell plasma membrane is then known as the neurolemma. Okay, we have axolemma, but when the Schwann cell is covering, suppose this is the axon, and Schwann cell is covering, suppose like this from both directions. I'm drawing it like this because I can't draw in 3D. Okay, Schwann cell is covering like this. So the outer surface of the plasma membrane of the Schwann cell covering the axon will be known as neurolemma. So get the concept clear. Neurolemma is not the axolemma. That is the outermost plasma membrane surface of the Schwann cell which is covering an axon, not simply Schwann cell. The Schwann cell which is covering an axon. Same way, just like you can see the way I had explained to you, the oligodendrocytes cover and they then give rise to the formation of myelin sheath. Same way, almost the same way, the Schwann cell covers it and is known as neurolemma. Okay, and what it does, it shields the axon from contact with the interstitial fluid directly. Okay, now again, gap between the two Schwann cells will be known as internodes, or again the node of Ranvier, where the myelination is absent. Okay, one more difference between the one specific difference I'll tell you between the Schwann cell and the oligodendrocyte is a myelinating Schwann cell myelinates only one axon, whereas an oligodendrocyte, I told you, its processes can bind to more than one axon, means it can myelinate more than one axon or more than one neuron in the CNS. Okay. So one major difference as far as the competitive exam is concerned is uh, Schwann cell myelinates only one axon, means one Schwann cell will myelinate one axon, means one neuron, whereas oligodendrocyte, one oligodendrocyte can myelinate more than one axons slash, that is more than one neurons. Okay, unless the axon is having the collaterals. Okay, that means again one neuron, but more than one axon simply means more than one neuron. Okay, that is the main difference between an oligodendrocyte and you can say and a Schwann cell. Okay, so this is what you should be knowing properly. And once you can also think of neuronal responses to injuries, which is a very practical problem. Suppose a neuron is injured. Suppose you might have come across many people who have, you can say, might have had paralysis or some kind of nerve damage. So one of their muscles may not be working properly. Okay, they might be having muscle twitches, this, that, one side paralysis, hemiparalysis, quadriplegic, all four limbs are in the paralytic stage. So what is the problem? So what happens when a neuron is injured? Okay, what happens is it responds to injury in a very limited way, because I told you they do not have the centrioles. They are somewhat arrested in G0, sometimes they are arrested in the G2 stage. Okay, so in general, more repair is possible. Okay, what has been seen in research in the human body, more repair after injury is possible in PNS. Okay, compared to the CNS, means peripheral nervous system compared to the central nervous system. That has been seen mostly. So what is the issue here? The issue is, what you can see is, after an injury, the nisal bodies in the cell body of the neuron or the cyton, it will disperse and the nucleus, it moves away from the centralized location as the cell increases the rate of protein synthesis, because the protein synthesis has to be increased, because injury has occurred. So cell will know that I need to now synthesize more and more proteins in order to recover, right? More and more different proteins will be needed in order for the recovery to take place, right? Now if the neuron recovers and regains functions, it will regain its normal appearance, the thing is fine. But the key to recovery, okay, what happens is, appears to be events in the axon. So, like for example, if I say the pressure applied, you can say during a crushing injury produces a local decrease in blood flow and oxygen. Now, the affected axolemma, it then becomes completely dead. It can become unexcitable, means it is not able to conduct any electricity, action potential. Now, suppose if the pressure is then elevated after an hour or two, what happens is that neuron may recover within few weeks, okay? But more severe or prolonged pressure, suppose somebody has crushed your hand with a tire or something, then it may not produce the proper recovery because it may completely cut or completely break down the axon. It may completely detach the axon from that place. Okay, that can happen. So in the PNS, what happens? Peripheral nervous system, the Swan cell. Okay, they play a major role in, you can say, damage repair. So what happens is the axon, which is away from the injury, okay, it is known as distal and proximal. Proximal means close, distal means away. The shoulder is proximal to my body, my hand is distal to my body, okay. So the axon distal to the injury site sometimes can degenerate, okay. The Swan cells, they do not degenerate, okay. What happens is they proliferate and they form a solid cellular cord. Okay, solid cellular cord it is known as, which follows the path of the original axon. Now listen carefully. As the axon recovers, you can say it can grow into the site of the injury and the Swan cell may wrap around the axon. But the key thing here is, if the axon grows alongside the appropriate, you can say, cord, okay, or you can say the cellular cord which is perpetuated by the Swan cell, then what would happen is it may eventually reestablish its normal synaptic contacts. But if it stops growing or it wanders in some other direction away from the cord, okay, then what will happen? Normal function will never ever return. That is what happens in case of, you can say, neuronal damage. So I'll show you one thing how the Swan cell goes for, you can say, myelination. You will be amazed to see that image. I'll just show it to you. Share the screen. Just tell me if you can. Just wait. Okay, now you will be able to see the screen. Okay, I think you can see the screen now. Good evening, Samesh. Can you see the screen? I think you may be able to see the screen right now. Okay, Gayatri. So this is how the myelination happens. Okay, Pratham. So this is how the myelination takes place. Look at it very, very carefully. This is a real-time myelination. So it will first surround the portion of the axon within the groove of its cytoplasm. Okay. Then the Swan cell begins rotation. Okay, look at step number 2. It rotates around the axon. Okay, it rotates around the axon, keep on rotating, so it will elongate its plasma membrane. Okay, it is just rotating and causing more concentric circle, you can say, circle formation. Okay, and that is known as the tightly packing of the membrane, and this is then known as the myelin sheath, a thick multi-layered plasma membrane which is perpetuated by the Swan cells. Okay, so this is how the myelin sheath is generated. And one more thing I'll show you, just look here. You can see the fragmentation. This is the distal lump. Okay. Now you can see here the Swan cells, they form the cord. You can see here. It forms the cord which grows into the cut. You can see it forms the cord and grows into the cut. This is the cord of the proliferating Swan cell. Okay. And if the axon, okay, it grows in this direction of the cellular cord, you can see the cord is joining. Okay, if it grows in the cellular cord's direction, it may be able to, you can say, again re-establish the connection. Okay, that is what I was trying to tell you. It will again re-establish the connection then. So this is a very good diagram of how you can say. Now you can tell your friends that how after injury swan cells can make this process happen very quickly. Okay, but whereas this, since oligodendrocytes they cover more than one neuron, okay, so they are not able to simultaneously produce this cord of, you can say, cell. So cell cord or this neuronal cord cannot be produced by the oligodendrocytes that efficiently in the CNS compared to Schwann cells in the PNS, on account of Schwann cells being able to myelinate only one axon. Okay, that is why you can say in the PNS the recovery is much faster compared to your CNS. Okay, much faster. So then we can switch on to the other topic. Okay, after this we can switch on to the, you can say, membrane potential of a neuron. Very important thing. Okay, membrane potential of the neuron. Now, in our one of the classes I was explaining to you the resting membrane potential, right? You might be remembering that class we had on Zoom. So same way neurons, they have their resting membrane potential. The resting membrane potential for a neuron, okay, you know it very carefully. Resting membrane potential, that is the most important thing when it comes to neurology or neuroscience. Resting membrane potential for a neuron is minus 70 millivolt. Minus 90 it is for the cardiac muscle cell. Minus 85 it is for the skeletal muscle cell. Okay. Minus 70 it is for the specific neurons. When the resting wave potential is minus 70 millivolt means what? Just I'm repeating that explanation again. If you have a cell, suppose this is the neuronal cell body, this is a neuron. Now inside of the cell is minus 70 compared to outside means what? The negative charges, the net negative charge inside the cell is minus 70, or minus 70 charges which are more inside the neuron compared to the outside of the neuron. That is the meaning. You will be having negative charge here also, negative charge here also. But the net negative charge inside is minus 70. Okay. So negativity will be perpetuated by chlorine minus ions, positivity by sodium, magnesium, calcium, okay, potassium. Okay. So all these things they perpetuate the, or they keep on maintaining the minus 70. And once minus 70 is attained, once minus 70 potential difference is attained, that time there is no perpetuation of electricity, means the cell is at complete rest. Therefore it is known as resting membrane potential. Resting membrane potential, the cell is at complete rest. Okay, that is what is known as the resting membrane potential. Now, how is the resting membrane potential maintained at minus 70? That is $1 million question. That is the conceptual understanding that you must have. Number one is coming to the extracellular fluid, means fluid outside the neuron. Okay, or you can say intracellular fluid, also the cytosol. The difference between the intracellular fluid and extracellular fluid, that is what we should be talking about. Because how do we maintain the charge? We maintain the charge by what? By just changing the concentration of the ions in and out of the cell. Right? That's all we do. Extracellular fluid, intracellular fluid. We change the concentration of the ions intra and extracellularly, and we get the change in the concentration of charges. Right? So that is what we are doing electrically, right, to the cell. So the extracellular fluid we always know contain more amount of sodium and chlorine ions, where the cytosol contains high concentration of potassium, okay, and also negatively charged proteins. So one thing you should, I'll write it down for you. the extracellular fluid means fluid outside the cell contains high Na plus and Cl minus compared to high means what? Compared to inside of the cell, meaning that inside of the cell you will also have Na plus, but concentration of Na plus will be lower to the concentration compared with the concentration of Na plus which is outside the cell. Intracellular Na plus will always be lower than extracellular Na plus. Same way, extracellular chlorine minus will always be again higher than intracellular chlorine minus. One more thing, intracellular fluid means cytosol. Okay, intracellular fluid is what? Cytosol or cytoplasm. It contains high potassium and negatively charged proteins. What does it mean? Potassium ion concentration inside the cell is always more than potassium ion concentration outside the cell. And the concentration of proteins are always present more inside the cell because it is being produced inside the cell in the rough endoplasmic reticulum. So, and proteins are mostly negatively charged. Okay, once we have learned about biomolecule chapter that I taught you, so proteins are really negative in charge. Okay, so negatively charged proteins are always more inside the cell than the, you can say, concentration of proteins outside the cell. Okay, so always remember this fact. So this is what will decide the overall charge distribution inside and outside of the cell. This is the first point how we start to determine the charge distribution inside and outside the cell. Okay, so many books they write, okay, in your NCRT it is also written Na plus is there, but they don't mention who is more, who is less. They just mention Na plus, K plus, this and that they have mentioned, but they'll keep you in dark and directly they'll put the question regarding the resting membrane potential. Okay, so this is the concept. Okay, the concept is the concept that I am telling you. Okay, so understand this very carefully. Which charge is more, which charge is less. Okay, then coming on to the next point. The next point are cells are very selectively permeable. We are all aware of this fact. So if the plasma membrane, suppose, okay, were freely permeable, okay, the diffusion will keep on continuing until all the ions were evenly distributed across the membrane, right? And a state of equilibrium existed. But at even distribution, okay, does not occur because cells have selectively permeable membrane. That's why resting membrane potential is not zero, because selectively permeable membrane is present. So ions cannot freely cross the lipid bilayer because the lipid bilayer is what? Is non-polar. The plasma membrane of a neuron or a heart or a skeletal muscle cell is non-polar in nature, lipid bilayer. Even if it has phospholipids, it is not that polar in nature. If you have phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, phosphatidylinositol present in the plasma membrane, still it is not that highly, you can say, polar that it will allow the charged ions to get in or get out of the cell. Okay, so what happens is this lipid bilayer is non-polar. Second concept. So ions just cannot pass freely through it and you will not get zero inside and outside. Okay, 0 MV. So what happens is, in order for an ion to cross, in order for an ion to come into the cell or go out from the cell, in order for ion to have influx or efflux from the cell, what you will need is you will need some transporters or channels, some specified gates which are the integral membrane proteins, because proteins are charged particles. They will allow charged ions to come in or go out. Okay, concept number two, you will need some specified protein gates, and those gates are directly, you can say, specific for each type of ion, because sodium ion channel will not let potassium come in, potassium ion channel will not let the sodium come in. Always know this fact, okay, for sure. Now understand one more thing. In order for a current to pass through, you need some resistance, right? Otherwise, current will keep on flowing, right? There will be incessant current flow without resistance. So the passive processes acting across the plasma membrane involve both chemical and electrical gradients. Okay. Now we know that positive and negative charges attract one another, and if nothing separates them, the oppositely charged ions will do what? Will move together and eliminate the potential difference between them, right? So a movement of charge to eliminate the potential difference is then known as current, right or wrong? This is what we have learned in the current chapter in physics. The very basic of the current is the movement of the charge to eliminate potential difference. What you are doing, you are going from higher potential to lower potential to eliminate the potential difference. So first you have to establish the potential difference. In order to establish the potential difference, you must have some resistance. Now, who will provide the resistance? The resistance will be provided by the lipid bilayer. Okay, so this is the resistor. Okay, and then potential difference can be established. And once potential difference is established, this minus 70 is what? This is the potential difference. Okay, minus 70 here, that side is not minus 70, minus 70 it is inside. So there is difference in the potential. Okay, so then the potential, due to the potential difference, due to the difference in the potential, the current will start to flow. Okay, always remember. So now we will be talking about the intracellular concentration of potassium and compare it with the extracellular concentration of sodium and their movement. Okay, so this is the concept regarding the resistance and the potential difference, because if the resistance is zero, if you use the equation equal to IR, if this is zero, okay, so current will be, you can say, kept on flowing, right? So you cannot have resistance to become zero completely. Then the potential difference will become zero in the end, right? So either you will become the, you can say, you might have heard of superconductors. Either will become this or the current will stop flowing. Okay. So if the current keeps on flowing, what will happen? We will keep on going for stimulus. Suppose I'm punching and I'm at this state only. I have kept on punching, I cannot come back to rest. So this is not what evolution wants, right? So suppose you are dancing, so you are dancing, dancing, dancing unless you die, right? So you cannot control the current because current flow will cause the stimulus formation. I'm talking, this is current. I'm writing, this is current, right? I'm moving the hand because of my nerves, again current and my muscles. The heart is beating because of the current. So the current flow is incessant, the heart will stay in that depolarized state and it cannot beat again. The neuron will stay depolarized or completely discharged. It may become then hyperpolarized and then it will never come back to its original condition, original state, and next action potential cannot begin and you will be in the state of epilepsy or fits. That is why fits occur, or you can say convulsions. They are all electrical disturbances, too much discharge or depolarization. So that is not what we want from the body, right? Otherwise the existence will become extremely difficult. You have to always then take the help of medicines. That is why this resistance is very, very significant, which is provided by the plasma membrane itself, right? Now talking about the potassium, so the intracellular concentration of potassium ions is relatively high, which I have told you. Now, whereas the extracellular concentration of the potassium ion is extremely low. Now, what will happen? The chemical gradient for potassium ions will tend to drive it out of the cell or not. Just think like this. This is the cell. Now, we'll talk about electrochemical gradient pertaining to potassium and sodium, because these two are the major ions which cause current flow. Electrochemical gradient. I should be saying gradients. Okay. First, potassium. So potassium is low here, is high here. So more potassium here, less potassium that side. So chemical gradient will do what? Will try to take it outside the cell in favor of its concentration gradient from high concentration or higher concentration to lower concentration. So that is what the chemical gradient will do. This is the chemical gradient. Okay, chemical gradient. Now, talking about the electrical gradient. Okay, so I can say, however, the electrical gradient opposes this movement. How? Because K plus inside and outside of the cell are attracted to the negative charges on the inside of the plasma membrane, right? On the inside of the plasma membrane. Listen to this sentence very carefully. And also are repelled by the positive charges on the outside of the plasma membrane or not, because Na plus is a lot outside. So charge wise, when I'm talking about electrical gradient means charge only. Now you will consider either plus or minus, only charge. So a positive charge is coming out. Won't this positive charge repel it? And we have inside the cell negatively charged, you can say, proteins. So positively charged, the positive will attract it. So it will not let the positive go out. So the electrical gradient, okay, it will repel the, or it will oppose the outflow or efflux of the potassium. But what happens is the chemical gradient is too much greater than the electrical gradient. Okay, the chemical gradient always wins because this is the concentration of ions, like the chemistry wins here. So the chemical gradient overpowers this little attraction and repulsion theory. And then, like in physics, you draw vectors, right? So outside vector may be like this, repulsion vector may be like this. So the resultant may be, this may be the resultant, okay? But potassium will come out. So this is your chemical gradient, this is your electrical gradient, this is the total gradient, right, which is towards the outside. Okay, so vectorially you can see we can represent the potassium electrochemical gradient like this, where the chemical gradient is, you can say, favoring the potassium ions to go out of the cell, and the electrical gradient is not favoring potassium ions to go out of the cell. But since chemical gradient overpasses electrical gradient, so in the end potassium will definitely go out of the cell. Okay, that is the benchmark here. Then we talk about the electrical and chemical gradient of sodium. Okay, now let's talk about the sodium electrochemical gradient. Now you will see again a very good theory. Now sodium is less inside the cell, sodium is more inside the cell. Sorry, more outside the cell, less inside the cell. So chemical gradient directly very easy. It will follow sodium to go for influx into the cell, means from higher concentration to lower concentration, just like your potassium gradient. But electrical gradient is just the opposite of potassium's electrical gradient. Why? Because you can say negative charges of protein are inside, so it will attract the positively charged sodium ions. Okay, and also inside, you can say outside plus charge is more, inside plus charge is less. Again, repulsion wouldn't be that great, and this potassium, okay, the sodium is too high, so it will come down its electrical gradient. So here both the gradients are causing the sodium to come in, because mostly due to the inside of the, you can say, cell membrane possesses the negative ions. Okay, so it will attract, whereas since it was inside, so it was attracting this and causing it to be opposed. But here this is attracting the sodium since this is coming from outside. Okay, so this attraction just due to the change in direction causes in the favoring, or you can say favorable electrical gradient to be produced for sodium. So sodium vector will be like this, chemical gradient like this, electrical gradient is also like this. resulting is a bigger arrow. Total gradient will be like this. So sodium influx is a very, very favorable process, okay, compared to potassium efflux. Okay, so this is the concept regarding sodium and potassium, how it comes in and goes out of the cell. A very easy concept. Okay, electrical and chemical gradient, when you combine, you get electrochemical gradient. Okay, so the word electrochemical gradient is given in all the books, but they don't explain the electrochemical gradient. So that students, what they do is they become very, very confused. In this way, you will never stay any, or you will never have any kind of perplexion or confusion with you. It will be all crystal, you can say crystal clear. So any doubt here in the gradient part? I don't think any doubt will be there, but you can clarify if you have any doubt in the potassium and, you can say, chlorine, sorry, potassium and sodium electrochemical gradient conceptual understanding. Any query, doubt? Okay, Gayatri. Okay, good, Pratham. So now coming on to one more thing, the resting membrane potential. Now suppose if the plasma membrane were freely permeable to K plus, that is a capital If, IF means it is not, but suppose assumption is plasma membrane were freely permeable to K plus, what would have happened then? Okay. So suppose if the plasma membrane were freely permeable, if the plasma membrane were freely permeable to K plus, but you can say impermeable to other positively charged ions. This is the case I am causing. It is not like that, but we are supposing this would have happened, but impermeable to other positively charged ions. Then what would have happened? What would have happened is, you can say, the potassium ions would continue to leave the cell until the electrical gradient which opposes the exit of K plus from the cell was as strong, okay, was as strong as the chemical gradient which drives K plus out of the cell. Okay, that would have happened because it is impermeable to positively charged ions. So positively charged ions would keep on coming here, and K plus would keep on coming here, so it would increase positively charged ions. So a time would come that even if K plus is higher here, K plus is lower here, okay, due to the excess accumulation of positively charged ions outside the cell, the electrical gradient would eventually become that high that it will become equal, you can say, to the chemical gradient eventually. Okay, and at that point in time, what would have happened is the membrane potential would have been reached at which there is no net movement of a particular ion across the plasma membrane, and that is the membrane potential we are trying to achieve or not. I am saying at minus 70 millivolt there is no more movement of, you can say, ions. So is this not the membrane potential at which no movement is happening? Now potassium cannot move, right? This much, suppose if this is the condition, so this has happened. Now no potassium can move out, right? So this is known as something known as equilibrium potential or resting membrane potential. Okay, so for potassium ion individually, okay, the equilibrium occurs at about the potential of minus 90 millivolt. So for potassium ions, minus 90 MV is the equilibrium potential. Okay, and the resting membrane potential of the neurons is minus 70 millivolt, which is a value very close to the equilibrium potential of K plus. So this minus 20 difference is due to, primarily due to the, you can say, the presence of Na plus leakage channel, because Na plus continuously keeps on coming into the cell, okay, via leakage channels. So there are specific leakage channels like this which keep the positively charged Na plus coming in. Okay, so what we have is the real value is minus 70 because of this only. Otherwise, it would have been equal to the equilibrium potential or resting membrane potential for potassium ion. Okay, this would have been equal to that. But it is not impermeable to positively charged ions because Na plus keeps on coming in through the leak channels, you can say. Leak channels are always open, okay, to that specific ion, regardless of the situation. If it is higher in concentration, it will let it come through its concentration gradient, always open, like that. So this is why the membrane potential is minus 70. Okay, so that is how I gave you the answer why the resting membrane potential is minus 70. Okay, most people will tell you neurons resting you can say resting membrane potential is minus 70, but they will not tell you why. So in biology, everything has a reason. If it is written minus 70, this is the reason. Because if we assume like that, after a point in time, the chemical gradient will become so high, it will oppose the electrical gradient will become so high, it will oppose the chemical gradient and sodium ions cannot come out of the cell because too much potassium, sorry, the potassium ions cannot come out of the cell because too much potassium has got accumulated outside, so plus will repel plus in such a way that no plus can come out. Okay, and that is minus 70, but sorry, that is minus 90 in case of a neuron for potassium, but it is minus 70 in case of a neuron generally because some positive comes in, that is Na plus channel through the leak channel, so there is this 20 difference. So we take this minus 70 millivolt as the resting membrane potential where no ion is moving in or out of the cell. This is the potential difference. Okay, at which no ion moves inside or outside the cell. That's why no current is being propagated at that specific potential difference. Okay, or that specific potential minus 70 millivolt inside the neuron, and therefore no current is propagated, and then we can say that a neuron is said to be in the state of rest when the inside potential or the intracellular potential of the neuron is minus 70 millivolt. Okay, so I hope this is completely clear, right? Any doubts here that why is it minus 70? Okay, any doubts? Why is it minus 70? Minus 70 millivolt, and what would have happened and how it stays minus 70, and how and what is the meaning of resting membrane potential, that no ion moves in or outside the cell. Okay, so this is the concept that I want you to know. Good. So if you have, since you have understood the concept, now no question, even if it comes from NEET, it comes from board examination, home examination, you can nail questions like this easily now. Then this was the toughest thing, and now in the next class we will do the complete action potential graph, right? So okay, then thanks a lot, and you can ask your queries on my personal number as always, and we'll be taking the or we'll be meeting next time. Okay, then thanks a lot. Let's see you in the